topic will be eukaryotic transcription. I've divided this into 3 mini presentations. The first is L10a, which principles of transcription, and we talk about RNA polymerases and assembly at gene promoters. L10b, we'll talk about processing of RNA polymerase II transcripts and the expression of mRNA. And under L10c, we'll talk about eukaryotic RNA polymerases and transcription, and the basal transcription apparatus, transcriptional activation, an important example of transcriptional control, the phosphoenol pyruvate carboxykinase gene. Then we'll talk about ribosomal genes and class 3 genes, which are tRNAs and 5S small ribosomal RNAs. And then finally, we'll talk about microRNA. We've already seen the central dogma of molecular biology, where we talk about the flow of biological material from DNA to RNA to protein. And again, here's our double-stranded DNA, and we've already talked about replication of DNA, and now we talk about the transcription of DNA into complementary RNA. So one of these strands is copied to make our mRNA. And then our next topic will be translation of RNA into protein. Over here's our double-stranded DNA, 5' to 3', 3' to 5'. We make an mRNA and we copy the 3' to 5' strand. That's called the template strand. And this gives us a sequence that corresponds to the sequence on the 5' to 3' DNA strand. We'll find that this sequence on the DNA may contain more nucleotides than the sequence in the mRNA. Because of our genetic code, we can divide this up into triplets, and the triplets code for particular amino acids, and 5' to 3' gives us a coding sequence. 5' in mRNA to 3' gives us a coding sequence. And then when we translate these on ribosomes, the first amino acids that are synthesized from the first codon will be at the amino terminal. So we synthesize from the amino terminal to the carboxy terminal. So this strand and this strand and this strand are in register and sequence with each other. This sequence represents an amino to carboxy terminus. Principles of transcription. Here's our DNA template. Notice it's 3' to 5'. Here our RNA polymerases polymerize 5' to 3', just like our DNA polymerases. Here's an incoming ribonucleotide triphosphate. Notice it's not deoxyribose, it's ribonucleotide. And these are our activated intermediates. The 3' group of the 5' to 3' existing strand attacks the alpha phosphate, and that gives us a phosphodiester bond, and now our 5' to 3' is 1 nucleotide longer. When we attack here, we release a pyrophosphate, and when we hydrolyze the pyrophosphate, that basically makes the reaction irreversible. Here's a crude picture of an RNA polymerase. We're going to have what's called a coding strand, and then we're going to have the template strand, and we're going to give these some other names. They're called several different things interchangeably. So this is our DNA strand that's being copied. Here's our RNA strand that's being made. This is the direction of the transcription, and we're adding to this 3' end. This is the 3' end. This is the 5' end. We actually started the sequence with the nucleotide that we have here, and it started about right here. But as this moves, the double helix reforms on the back side. The double helix is being separated on the front side. So this is the 5' end that was synthesized first, and we're synthesizing towards the 3' end. And as the polymerase moves more to the right, this ribbon that's coming off, the RNA ribbon, will get longer and longer and longer. Turns out the DNA and RNA can only base pair with each other for about 10 nucleotides before they split off. So this strand coming off here represents the 5' end that no longer will base pair with the DNA. The coding strand is also called the sense strand. So the sequence of the coding strand makes sense according to the genetic code. So the triplets make sense according to the genetic code. The template strand is also called the antisense strand. So it's the complementary strand of the sense strand, and it's the template strand that's transcribed. So here we have the DNA non-template coding strand, which is also called the sense strand. So this is the sequence. The DNA template strand, which is complementary, GC, 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 TA, AT, AT, etc. And then our RNA transcript was transcribed from this strand, but it will have the same sense as the 5' to 3'. So this is our product is 5' to 3', the sense is 5' to 3', and the only difference is we replace T's with U's. Remember, RNA contains uridine, DNA contains thymidine. So here's our RNA transcript. Here's a nice representation of RNA polymerase. Sectional view of an RNA polymerase. So it's been sliced along the plane of our DNA. And notice we have to open the DNA in order for the antisense strand to be transcribed. So this shows where we're taking the DNA apart. And when we do the unwinding, we have positive supercoiling. And when the RNA polymerase moves through the transcription bubble, we have rewinding. So we unwind in front of the RNA polymerase. We rewind after the RNA polymerase. And after we're finished doing this, there's no change in the supercoiling state of the DNA. So here we're changing the supercoiling state. This reestablishes the supercoiling state of the DNA that we started with. Here's the active site of the RNA polymerase. This is RNA-DNA hybrids. Remember, we base pair for about 10 nucleotides before our base pairing breaks. This was the first part of the RNA we made. This is the RNA that's being extended. This is where the nucleotide triphosphates, they actually come into this channel. So quite an elegant molecule. Notice we have what looks like a bump. Since this is a cross section, this is actually like a pillar that's in the middle of the enzyme, and it keeps the two DNA strands from reassociating. So really a nice picture of an RNA polymerase. This is the picture that we saw before, although we saw this with DNA polymerase, but this is basically the same picture. The mechanism is the same for a DNA polymerase and an RNA polymerase. So the incoming nucleotide triphosphate is attacked at the alpha phosphate. So here's our incoming nucleotide triphosphate. Here's our template strand. Here's base pairing. So this is where we get recognition, base specificity, sequence specificity. Here's our sequence specificity. The 3-hydroxy group is actually shown as an oxyanion, negatively charged. That makes it even a stronger nucleophile, and it attacks this phosphate group. Here's the electron pair moves over here, and this is the pyrophosphate that leaves. Again, magnesium and aspartates, which make chelates with the magnesium. They align the triphosphates. The base pairing aligns the ribose. So if you go back and look at DNA polymerase, this is exactly what we saw with DNA polymerase. And now, upon this nucleophilic attack, we've made a phosphodiester bond. This is a 3' to 5' phosphodiester bond. Here's our next 3' OH that we'll add the next nucleotide to. So the incoming is attacked at the alpha phosphate by the 3' hydroxyl of the growing RNA chain. So we have 4 RNA polymerases that transcribe the human genome. RNA polymerase 1 transcribes the 18S, 28S, and the 5.8S ribosomal RNAs. It is found in the nucleolus. RNA polymerase 2 transcribes mRNAs, microRNAs, MI is for microRNAs, and most of the small nuclear RNAs. And we'll find out about small nuclear RNAs here in L10b. And RNA polymerase 3 transcribes tRNAs and 5S RNAs and several other small RNAs. And then mitochondrial RNA polymerase transcribes mitochondrial RNAs. So here's our 4 RNA polymerases. 1 through 3 work in the nucleus. The 4th one is found in mitochondria. Now we're going to talk about RNA polymerase 2, which is the polymerase that makes our messenger RNAs. So here's polymerase 2, 12 subunits, very small to very large. It catalyzes the RNA synthesis. TPB, that's called the Tata box binding protein, and it's going to recognize a sequence in front of the gene that's going to position the RNA polymerase before it starts transcription. Here's transcription factor 2A, transcription factor 2B, transcription factor 2EF, transcription factor 2H. And then we have elongation factors. Once we assemble this huge complex, the RNA polymerase starts to transcribe, and then at that point it uses what are called elongation factors, which substitute for some of these proteins. So we'll get some more detail on these as we put the RNA polymerases together. So here's a DNA polymerase II promoter. So we have promoters like this in front of most of the genes that code for protein sequences. So we have various regulatory sequences. Minus means this is in front of the start site rather than down from the start site. And this is called the TATA box, TATAAA, and this is found in a large number of promoters, although there's about as many promoters that don't have TATA. And here is another sequence called the INR sequence. And if you don't have a TATA box, you have to have one of these in order for the RNA polymerase to bind. So you can bind the RNA polymerase without the TATA box, but you have to have this sequence in order for it to work. So here's our 5' end, here's our 3' end, and now we're going to use the information we saw with the different subunits and the information on this promoter, and we're going to assemble the RNA polymerase on a DNA sequence. So first, here's the TATA box binding protein. This sequence in here of DNA has TATAAA, as we saw earlier. This is transcription factor 2B, transcription factor 2A. And notice 2A and 2B are on either side of the TATA box binding protein. And also notice that when TATA box binding protein, and I'll call it TBP, when TBP binds the DNA, notice what it does to the DNA. It changes its direction. It basically puts a kink in the DNA. So it changes the structure of the double-stranded DNA, and it's flanked on one side by transcription factor 2B and on the other side by transcription factor 2A. And we would be moving from right to left. So +1, this is where transcription starts. This is at about -30 nucleotides. So here's our +1. At this point, the RNA polymerase would start transcribing our template strand. So the assembly of this particular complex helps recruit the rest of those subunits and the RNA polymerase II that we talked about. So now the next slide will show the sequence in more detail. So what we're going to make is something that's called the basal transcription apparatus. So here's our TATA box at -30. Here's our INR sequence at +1. The TATA box and our transcription factor 2A and transcription factor 2B. These three factors bind. TATA box binding protein is binding here. Transcription factor A would be on this side. Transcription factor 2B would be on this side. Remember how they flanked? And now we bind transcription factor 3F and the polymerase II. It binds to this complex. And then we bind E and H, and this is what we get. This is our double-stranded DNA. This is transcription factor 2B. Here's transcription factor 2A. Here's a TATA box, the binding protein. And here's F, E, transcription factor 3H. And this is the RNA polymerase. This is called the closed complex because we haven't separated the DNA strands and made our transcription bubble. Now, DNA unwinding to produce the open complex. So we call this the open complex because now we've separated our DNA strands and we can transcribe our template into mRNA. Phosphorylation of polymerase II, initiation and promoter escape. So if we notice, polymerase II has a protrusion on the 5' end. If we call this the 5' and this the 3', it has a protrusion pointing towards the 5' end, and this protrusion gets phosphorylated, and this is called the C-terminal domain of RNA polymerase II. So this is the C-terminus, and the C-terminus gets phosphate groups. We can see the phosphate groups down here. So we're going to phosphorylate the C-terminal domain. And then our initiation factors and elongation factors are going to bind, and they're going to separate the RNA polymerase from TF2A, TF2B, and the Tata box binding protein, and you can start to synthesize the RNA. Notice, with the synthesis of RNA, and this is different from DNA polymerases, you don't need a primer. All you need is a complementary strand. So RNA polymerization doesn't need a primer. And if you think about that, you already had insight, because when we made the RNA primer in our DNA polymerase, we synthesized an RNA polynucleotide without a pre-existing primer. So you can synthesize RNA without a primer, but you can't synthesize DNA without a 3' OH. So we phosphorylated the C-terminal domain. We start our initiation, and the polymerase releases from the Tata box binding protein and TF2B and TF2A. And once this gets far enough downstream, another RNA polymerase can bind to this complex. So we can have multiple RNA polymerases on one piece of DNA, on one gene, making mRNAs. So here's our elongation factors. We get elongation. Eventually we come to sequences that signal termination. The RNA dissociates. Pol2 releases, and it becomes dephosphorylated, and then it can bind again. This is the Tata box binding protein binding to the DNA, and notice how it's putting a loop into the DNA. Remember how it made the DNA change direction? Well, this shows that it actually morphs around the double-stranded DNA. Here's our entering DNA. This is called downstream. Transcribed DNA upstream. Here's our active site. Here's the RNA leaving via exit. Here we would have the nucleotide triphosphate. It's going to be bound by the magnesiums, the triphosphate by the magnesiums. The base part will be base pairing with this DNA. And as we add nucleotides, the RNA polymerase moves along the DNA molecule. Remember, it's unwinding it in this direction. It winds it back when the DNA leaves. So here's the RNA polymerase 2. Really nice series of pictures.